Okay, so we are starting on our final unit, thank God. We're going to go through chapters 14, 15, 16, and 17. And because of a lot of the material overlapped in 14 and 15, that's why I put them together in a single PowerPoint. However, you will have individual mastering homeworks for all 4 chapters. So in this chapter, we're going to talk about how microbes become established in humans and lead to infection and disease. And then we're going to finish up talking about epidemiology. I've also uploaded several handouts that I would have just given to you in class, but I wanted to make those available too because those have some useful information in them. So the first thing to remember, and this I think also with COVID-19 is what a lot of people don't understand, is that coming in contact with a pathogen and then having that pathogen be able to gain entry to the host and then also to go ahead and cause a disruption of health, it's really a continuum. Because your body surfaces are constantly exposed to microbes and you always have pathogens living in and on you, but you're not sick all the time. So it means that those pathogens are kept in low numbers and they can't cause an infection. However, at some point, you're going to reach a level where you are immunocompromised or susceptible enough so that a pathogen is going to be able to penetrate your host defenses, get into the tissues and start multiplying. And that is what we refer to as an infection. Okay, so infection comes first. And then when tissues and organs are disrupted, that is what we call a disease. Okay, so the infection comes first, and then infection, if it's allowed to penetrate far enough, that leads to disease. And obviously, what we're talking about is infectious disease, meaning that it's either a microbe itself that's causing tissue damage, or it's something being secreted by the microbe, such as a toxin. So we mentioned this term a lot before too, normal flora, normal biota, resident biota. What that means is that you have, and the number I gave you at the beginning of class was up to 10 times the number of microbes living in and on your body as you do cells that make up your body. Okay, so you're absolutely covered with bacteria. A lot of those bacteria are infected with viruses. You have fungal cells, yeast cells. So yeah, you're covered. Okay, and that's what we refer to as normal or resident biota, meaning that they're pretty much there all the time. Okay, and this is all of your mucous membranes, your skin, your intestines, your gut flora. So actually, there is a research project, and the big research project previous to this was the Human Genome Project, where scientists work together all over the world to sequence all of the A's, G's, C's, and T's in the entire human genome, all 3 billion base pairs. Now, the next project is called the Human Microbiome Project, or as I like to call it, poops from around the world, because basically what they're doing is looking at gut bacteria and seeing what people who live in different regions, in different continents, have in terms of different species in their intestines, because gut health is incredibly important in terms of prevention and also maintaining health. So, for example, there are regions in Africa that have little or no asthma or allergies. And so is there something different about their gut flora that makes them not susceptible to those types of infections as compared to, say, the United States? Okay, so that's what that project is. So your body is kind of like an ecosystem, and it has a wide range of habitats, and you're going to support a wide range of microbes. So here are sites that are going to have bacteria, viruses, yeast cells. So anything on your skin and contiguous mucous membranes, meaning, your eyes, your nostrils, urogenital regions, your upper airway, your mouth. all of those are supposed to have lots of bacteria, right? Because we're breathing all the time, we're touching things, we're in contact with the external environment. So these, probably make sense to you. So, not the upper urethra, but the outer opening, external genitalia, your external ear, external eyes, such as your eyelids, conjunctiva, all of that, if you sampled from it, we did, a swab and put it on an agar plate, yeah, you'd have a bunch of bacteria growing, and that would be completely and totally normal. This is actually what we wanted to show you in lab 16, so it's kind of unfortunate that we weren't able to do that. So here are some fluids and body parts that you would expect to not have any microbes in them. And a lot of these, I think, make common sense too. So, your blood normally should not have any microbes. Urine, when it's in the kidneys, the ureters, and the bladder, your cerebrospinal fluid. Anything before, it enters an orifice that's open to the environment. So yeah, should you have bacteria and viruses in your bones, your pancreas, your lungs? No, you shouldn't. Okay, and when I say lungs, I mean deep in the lungs. We're not talking about the upper airway here. The one thing that surprised me on this list was actually your sinuses are supposed to be, basically close to being sterile. So you're not supposed to have a lot of normal flora in there, and that's why people, a lot of people struggle with sinus infections. So the term biota and flora mean the exact thing. Now, one thing that's interesting, and also in lab 16, excuse me, gotta swig the bang, is that your normal flora constantly changes. So, if you're feeling sick, if you have, let's say, a case of influenza, and we were to take, samples of your upper airway, it would look different than when you're all healed and feeling better. It also fluctuates with hygiene, diet, hormones, such as you see in pregnancy or menopause. If you're on an antibiotic, things are going to look different. So remember the key term here, and we're going to carry this over. I want you to know it also for this chapter, is the term microbial antagonism. And what that means is that we want all of those species of normal flora fighting against each other for real estate and for nutrients. Because if they're all fighting, then one of them cannot take over and kind of, grow over the other species that are there. So we talked about this in the drug chapter. And if you remember the term super infection, that's when you take an antibiotic and it wipes out the bacteria. Because of course the antibiotic can't distinguish between the good guys and the bad guys, and then you end up, let's say, with a yeast infection or colitis in the gut. So we want microbial antagonism. That is a good thing. And unfortunately, people who have compromised immune systems, so, namely the very young, the very old, or people who are battling a lot of other, comorbidities, such as cancer, hypertension, diabetes, those types of things, those people are especially susceptible to what we term endogenous infections. And an endogenous infection is one where the microbe is already in or on you, but now somehow it has overcome microbial antagonism and it starts to overgrow. So, for example, super infections would be endogenous infections, okay? When a female takes antibiotics for a sinus infection or whatever and then ends up with a vaginal yeast infection, the yeast is already there. So that's an example of an endogenous infection. And we have, quite a few terms for it. So super infection is another one. But also opportunistic infection, secondary infection, they can all mean the same thing, okay? But the key is an endogenous infection is when the microbe causing it was already there to begin with, in or on the person. So how do we pick up our normal flora? So ideally, every single birth that occurred would be a vaginal birth, because the baby gets seeded with normal bacteria from the birth canal and from mom. Of course, that's not a reality. There certainly C-sections can be absolutely necessary. So unfortunately, the newborn bypasses getting seeded. Also, if mom has syphilis or she has HIV, then you don't want a vaginal birth because you don't want the baby to get seeded with those known pathogens. But it's basically, having caregivers close to the infant. So when a baby's born, remember, they basically, they have very little functioning immune system. So, it always kind of freaks me out when I see like a tiny, tiny newborn on a plane or out at the mall, because I just think, oh my God, what potential pathogens are out there that could potentially infect that baby. So it's usually, the parents, either mom, whether bottle feeding or breastfeeding, just holding the baby, being close to the baby, and then taking the baby to a pediatrician. I mean, that's how infants slowly get seeded with their own normal microflora. And they tend to, kids that are in the same family, the parents and the kids tend to have pretty similar normal flora because they're all living together in the same environment. So when we talk about how microbes are, and specifically, remember, we're talking about pathogens, okay? So 99.9% of bacteria are, commensals. They live in or on you, and either they're doing you a favor or they're just there and they don't bother you. But when we have a pathogen, that is when that organism is a parasite, okay? And they're going to start to grow. They're going to use your nutrition and basically live like a parasite off of you. So there's a lot of factors that go into this, okay? One, if you look at here, and this is what we're seeing, especially with COVID-19, the condition of the host. And, it's true that younger people could potentially just be carriers of COVID-19, but they're not going to get the actual disease. However, they could pass it on to, say, their grandparents, or if there's a newborn baby in the house, or someone who's more immunocompromised. So a lot of whether a person gets sick depends on the condition of that person. And, for example, let's say you and a friend are standing right by each other, someone happens to sneeze and they have a rhinovirus, and you both get the same infectious dose or amount of rhinovirus. You might get a cold, but the person standing next to you may not. And there's a lot of factors, okay? Maybe you are feeling stress. Maybe you had a sinus infection going on already, and so that kind of lowered your immune response. Maybe genetically, your immune system is a little bit weaker, and so it tends to attack people who, for whatever reason, at that point in time, are immunocompromised. And we're all immunocompromised to a certain extent, and I think certainly right now, more so because, we're under quarantine and there's all these economic and social pressures, and, so certainly, we tend to be more immunocompromised due to stress. Okay, so that's one thing. It's the condition of the person who's receiving the pathogen. The other thing is in the organism itself, and we refer to that as its pathogenicity or its virulence. And pathogenicity just means how likely is it to cause disease. So the higher the pathogenicity or virulence of the organism, the more likely it is to cause disease, and you need to receive it probably in lower levels because it hasn't developed ways to get inside the host to gain access to the host cells. Okay, so highly virulent organisms are more dangerous, are more easily spread, and are able to cause disease in a wider range of people, no matter what their health condition. So COVID-19 is pretty virulent, okay? It's highly transmissible. We are seeing younger people of a lot of your age categories, unfortunately, getting sick. Now, they usually recover, okay? But the fact that they're getting sick in the first place, but yet they're young with mostly healthy immune systems, tells us that this is a highly virulent organism. So, pathogenicity, remember, equals virulence, okay? And it simply means an organism's potential to cause infection or to cause disease. We can then divide organisms into two different categories, okay? So we have true pathogens, and then we have opportunistic pathogens. Opportunistic pathogens are organisms like yeast, let's say Staph aureus, E. coli, organisms that are usually just part of our normal flora. But then when things get disrupted, because microbial antagonism isn't working, they can cause endogenous infections. Okay, so basically they're already there in and on you, but something happens and now they're able to overgrow and cause an opportunistic infection or a super infection. Okay, and they are always endogenous infections. True pathogens come from the outside. Okay, so Ebola and a lot of viruses fit into this. There's really no good reason. Viruses they're never just there and doing nice things. Okay, they're usually there to cause infections and to replicate themselves and destroy cells. That's what they do. So if we look at, and certainly I need to update this slide because COVID-19 would fit in here, but whether we're talking about Ebola, Zika, and then any organism, like the organism that causes syphilis or causes gonorrhea, they are true pathogens. It's basically their only purpose to exist in this world is to cause disease. And these are organisms that don't hang around on us normally, and so they are coming from the outside, and that's why we call them exogenous infections. Okay. So I'm about at 17 minutes. I'm going to go ahead and stop here and get this uploading, and we'll pick up here. Thank you.